Hey, this is a video lesson today. I'm taking you through gas exchange, the final part of the booklet, which is in the leaf. Okay, so what I'd like you to have a go at first of all is to pause the video, okay, and to explain how tissue fluid is formed and how it returns to the circulatory system. Okay, so we're going to have a look at someone else's answer in a minute as well and compare your own. So stop the video and have a go. Right, you should be back with us now then, having done your starter. Okay, so let's have a look at this answer and do a comparison. So at the arterial end of the capillary, the hydrostatic pressure is high, forcing out fluid. Okay, so that's going to be underlined in the exam. It has to be the word fluid. They don't want water, they don't want plasma. Both of them are incorrect. Okay, so make sure that you've got fluid there. And if you want a little bit more detail, that will contain soluble molecules that the cells need. Okay, that's not required, that's why it's in brackets. And that forces the fluid out of the capillary, out of the blood into the tissues. So you do have to have it into the tissues. So that's your first mark point. However, the plasma proteins remain in the lumen, so this is within the capillary, would be fine there as well, as they are too large to cross the capillary wall. So that's your second mark point. Plasma proteins remain in the blood because they are too large to pass across the capillary wall. Third mark point, these proteins, the plasma proteins, the proteins in the plasma, lower the water potential. Okay, so that's really important that you're talking about water potential. In a lot of the spot tests, it was all about osmotic pressure, which is what the water potential gradient can be measured as. Water potential gradient can be measured as an osmotic pressure. But because of the new mark schemes, it's better to stick with water potential. Okay, so that'd be a little bit better. When the hydrostatic pressure is dropped due to volume loss and friction at the venous end of the capillary, water moves back into the plasma, so back into the blood or back into the capillary by osmosis. The reason is because there is a water potential gradient. Okay, so water potential gradient between the tissue fluid and the inside of the capillary because of the plasma proteins being there present. There's always more tissue fluid formed than there is reabsorbed at the venous end, so the excess is absorbed by the lymphatic system, so that's your final mark point there. Okay, so your answer should be as full and detailed as that, maybe even more so about the water potential gradient, so saying that the water potential is lower inside the capillary because of the plasma proteins lowering the water potential. Therefore, there is always a water potential gradient from the tissue to the blood for water to move by osmosis. However, it only does this at the venous end because the hydrostatic pressure drops below that opposing osmotic pressure, that opposing water potential gradient. Right, so I wanted to recap that because in the spot test, we didn't get all those key points and we're missing a bit of that detail. And the other thing that I wanted to cover was talking about the plasma proteins and why we swell when we're starving. Okay, so that important statement there that the proteins in the plasma lower the water potential, if they weren't there, you wouldn't have a lower water potential. So this water potential gradient that is produced to pull the water back by osmosis is not there. Therefore, fluid would not be reabsorbed, water would not come back by osmosis. There'd be far more excess than the lymphatic system could absorb, and it would remain in the tissues and cause swelling. Okay, so today we're going to do a bit of recap of what we've done so far, and then we're going to do our final specialised gas exchange system, which surprisingly is the leaf. Okay, so we're going A, B, C, D. In class we do it with our cards, but today you'll just have to decide which one you would do. I'll give you some time to think. Okay, so here we've got a graph. Oxygen uptake per gram of body mass and body mass. Okay, you can see shrew, very small animal, very high oxygen demand, very low mass. Elephant, high mass, lower oxygen demand. Okay, so Which of those organisms has the smallest surface area to volume ratio? Okay, so the smallest would have been the largest organism here at the top. So it's not losing a lot of heat across its surface, therefore it doesn't need to respire to replace that heat as much, and so it doesn't require as much oxygen for that respiration to replace the heat lost across its surface. That's why we've got that trend. Okay. In A, B and C boxes, we have the gas exchange systems of insects, mammals and fish. Which is which? Time is up. This is the fish. You can see water moving in through the mouth and out across the gills. Mammal, in and then out. Insect, these are our tracheal branches. Okay. Which gas exchange system has the spiracles, trachea and the tracheoles? We should be picking straight away for that one, the insect. Oh, something's gone a little bit weird there. Oh, there we go. Okay, so this is a picture of a spiracle. Now the hairs around the spiracle reduce the loss of something from the surface of the insect, and they do that because they trap this around the hole. So what do you think it is? What would you be trying to reduce the loss of if you're an insect when you open your spiracle? Water, of course. Okay, so water droplets get trapped by those tiny little hairs. When the spiracle is opened, gas exchange occurs, but the water remains trapped by the water vapor gets trapped by the hairs, so that helps to reduce the water loss. Okay, now we did do this one last lesson and Tom picked correctly, so have a look, see whether you can pull it out again. This graph suggests that spiracles open in response to what? Carbon dioxide decrease, increase, oxygen decrease, oxygen increase. We're looking at the graph, they open when there is an increase in carbon dioxide. Okay, now you may have to look at this one for a little moment. So this is labelling 1, 2 and 3 in the diagram. So is it filament, arch, lamellae, or is it arch, lamellae, filament, or is it arch, filament, lamellae? If you pick C, you're right. So here we've got our gill arches. When we look at each one, it's made up of many filaments. And then each one of those filaments, when you look on top of it, has on top, like shark fins, lamellae sticking up. And you can see the counter current nicely in this diagram as well, where the water's flowing in the opposite direction to the blood flow in the capillaries at the centre of the lamellae. Okay, so which one of these is not correct? A counter current maintains the concentration gradient for gas exchange across the gill surface for the whole length of the capillary. Many lamellae covered in many tiny filaments give a large surface area for gas exchange. A parallel current maintains the concentration gradient for gas exchange across the gill along the whole capillary. Charlotte, I'm in the middle of videoing. Both lamellae and capillary cells are flat and thin and give a short diffusion distance for exchange of gases. Okay, so which one is not correct? Well, this one is not correct because many lamellae are not covered by many tiny filaments. The filaments are covered by lamellae, so they're the wrong way around. Okay, a parallel current does not maintain a concentration gradient, so this one is not correct. A counter current does maintain a concentration gradient for gas exchange across the whole gill surface, that one's correct. And both the lamellae and the capillary walls are flat and thin, giving a short diffusion distance, so that one's correct as well. Okay. So here's where I'd like you to stop the video and I'd like you to fill out the blanks in the boxes. Okay, and then when you think you've got everything, then you can join me again. Okay, so if we think about the fish then, How does it have a large surface area? So in this box here, you should have, it's got many filaments, each with many lamellae, and also many capillaries. So check that. Have you got, has many filaments, each with many lamellae and many capillaries there. Okay, that's how we've got a large surface area for exchange in fish. What about in insects? Well, in insects, we've just got many tracheals. So those breathing tubes, many branches to them. Okay, so the idea that there's many tracheals, many branches, that is the way that we've got a large surface area. It gives us many ends to the tracheals in contact with the tissues of the body of the insect. Okay, so then we have to work out what factor in Fick's law is missing here. So this one's talking about cells are always respiring and so producing carbon dioxide. Some insects are able to pump in and out of their bodies and replace them. Replace air saturated with carbon dioxide. So this is all about exchange and concentration gradients. So here would be maintain concentration gradients. Okay, so in insects it's mainly diffusion, so you could have written this as well for oxygen, so they're always respiring, so using oxygen, so it's always going to be moving by diffusion down the tracheals into the cells, whereas carbon dioxide is always being produced, so it's always going to diffuse out from the tissues to the tracheals. It's much more complicated in fish, so in here we're going to have lots more information. Okay, so in our fish column here, we're going to have ventilation of the gill. So that's water moving across the gill surface. We're going to have circulation of the blood through the capillaries and the lamellae. But then most importantly, there at the bottom, you should have countercurrents of blood and water, maintains a concentration gradient across the whole of the gill or the whole of the lamellae. So this bit here, ventilation of the gill is passing water over the gill surface. That's happening all the time. Circulation of blood is happening all the time. And also the fact that they are in a countercurrent with each other. So the blood and the water are in a countercurrent, maintaining a concentration gradient across the whole of the gill, the whole of the lamellae. And then finally, this is about having a thin diffusion distance. I can see that because in the fish they've got very thin lamellae walls, the blood is in close contact with the water, the walls of the capillary are 1 cell thick. In the insects, the diffusion is between the tracheoles and the body tissues. Okay, so there's a short distance there between, because the tracheoles go all the way to the body tissues. So if we can compare and contrast, then we would be saying similar things that are adaptations but in different ways. Okay, so what we're going to do now is we're going to have a look at the leaf. Now the leaf is an organ, it's obviously found in a plant, and its main job is photosynthesis. But to enable it to carry out photosynthesis, it must also exchange gases. So this is a little bit different to the other gas exchange systems, because in all the other gas exchange systems, we were looking at movement of oxygen from the air into the organism and carbon dioxide out. It's not quite the same with plants because the leaf is the site of photosynthesis, so gas exchange is for that process. So we might think then about when plants are photosynthesizing, when they're not photosynthesizing, and whether the exchange of gases changes. We're going to look at dicotyledonous plants. Okay, so this is the type of leaf that we've been asked to look at by the exam board. We're going to explain the adaptations of the leaf, again thinking about Fick's law. So how's it got a large surface area? How do you maintain a concentration gradient? And how is there a thin distance for exchange? And then describe the adaptations of a very specific type of plant that you've met at GCSE already, which are the xerophytes, the cacti. Okay, and then we might be able to then compare some exchange surfaces. So, gas exchange in the leaf of a plant. Well, all plant cells need oxygen and they need that for respiration. So you've got to be very careful here, okay? It's not that plants don't need oxygen for respiration, they do. And they need that all the time because they're always respiring, okay? And they also produce carbon dioxide during respiration, so they've still got that waste product as well, just like the rest of living organisms. But there is another process, okay, that plants need to do, and they also produce gases doing this, or produce a gas doing this. I've already spoken about it. It's photosynthesis. Now, in the leaf, that's the predominant process, if you think that's going on in a leaf in the daytime when the light is shining. And the gases that are used, it needs carbon dioxide and produces oxygen. So you can see the links here. So if a plant needs oxygen, but it produces oxygen, then it can use its oxygen that it produces for respiration. If it needs to get rid of waste carbon dioxide, well, it can actually use that in the process of photosynthesis. Like that. Okay. So it's all about balance in a plant. Gas exchange is about making sure that we've got enough carbon dioxide for photosynthesis, and of course we'll produce oxygen. Okay, so here's a couple of points to ponder. We'll come back to you later on. So during the night time, okay, levels of carbon dioxide around a leaf go much higher, and if you measure the carbon dioxide at different levels in a plant during the day, they're lowest at the leaf height. So we'll come back to those. They're the kind of difficult AO2 and AO3 questions they can ask in year 13. Okay, so here is your cross section through the leaf. You need a sketch of this really, so I'll pause the video now and sketch out this diagram. Once you've sketched it out, you can start to label it up. So the cuticle is what's found on the outside. Okay, just below that we've got the palisade cells. So these are the ones that you drew at GCSE. They're the ones that we drew when we did our basic plant cell structure, and their role is photosynthesis. You can see they're full of chloroplasts, packed out chloroplasts with chloroplasts. So that's their main role in this tissue. Sorry, in this organ. The reason we call it an organ is because it's lots of different types of tissue. So I'm naming different types of tissue working together now. Here is the xylem. So these are the very big tubes in this bundle here. Okay, and below that is the phloem. So these are the tubes that transport water, in the case of xylem, water and mineral ions around the plant, and for phloem, sugars. So actually from the leaf, the xylem is bringing water to this site because you need water for photosynthesis, and when the sugars are made, they're being taken away, taken to the roots and the other parts of the plant that need them by the phloem. So this is a transport vessel made up of all these tubes. Okay, and then you can see quite a predominant tissue within the leaf is this spongy mesophyll. Now its job is it's involved in gas exchange. So there are big air spaces between these cells, which allows gases to move in and out of the leaf. And then finally what we've got is the epidermis, which is the layer found at the top as well and the bottom here, just underneath the cuticle, and most importantly in gas exchange, some specialist cells guarding the stomatal pore or the stomata, which are the holes through which gases are exchanged with the leaf, and they're found under the underside of the leaf cell, the leaf organ. Okay, so now we need to think about why gases are exchanging and which ones are exchanging. Okay, so gases only exchange through the stomata. This is because the cuticle is very thick, okay, particularly on the upper leaf. It's a very thick waxy layer, okay, and that prevents evaporation of water through the leaf surface. But it also means diffusion of gases is very limited through that. So we have these holes which allow these stomata that open and close like spiracles do, and they allow gases to exchange. But now you can see that it's carbon dioxide that is moving in to the leaf and oxygen that's moving out. So we need to think about why that's happening. So if you think about the mesophyll cells, the palisade cells, they contain lots of chloroplasts. In the chloroplasts, photosynthesis is taking place and it requires carbon dioxide. Okay, so the chloroplast requires carbon dioxide and it produces oxygen. Obviously, these cells are still respiring, so they'll be using oxygen and producing carbon dioxide. But photosynthesis is the predominant process in terms of the balance, and so the gas that moves in is the one which is the lowest concentration. It's the one that's being used the most. So that's carbon dioxide being used up in photosynthesis, and oxygen moves out because it's a higher concentration in the air space because it's being produced by photosynthesis. Obviously, that's when photosynthesis is occurring really quickly, and that's the normal balance that we think about. Okay, so first question, a leaf is an organ. What is an organ? All right, so we said that we could refer to this as an organ. And I said it was because it was made up of lots of different types of tissues. So we've got palisade tissue, we've got spongy mesophyll tissue, we've got epidermis, we've got transport tissues here, xylem and phloem, and they're all working together for the leaf to photosynthesize, which is its role in the plant. Second question. Describe how carbon dioxide in the air outside a leaf reaches the mesophyll inside the leaf. Okay, so it's 3 marks. So it's outside of the leaf. Describe how it gets into this mesophyll cell here. So it's going to move through the stomata into the leaf through the stomata. So one of the mark points is the name, the pore. Why is it moving in? Well, it's diffusing simply down a concentration gradient, and what's producing the concentration gradient is the mesophyll cell is using the carbon dioxide in photosynthesis. So it's using the carbon dioxide in photosynthesis, therefore it's got a low concentration, lower than the air space, so carbon dioxide moves in. That lowers the carbon dioxide concentration in the air space below atmospheric concentration, and so carbon dioxide diffuses in through the open stomata. Okay, so you can see that there in the mark scheme. So the idea, it was a group of tissues. Carbon dioxide is entering via the stomata. It's an open stomata, so you can talk about that because the guard cells have opened it. It's diffusing through the air space, and a description of concentration gradient, if you like. So it's down a concentration gradient, so saying it's being used by photosynthesis in the cell and therefore is diffusing down a concentration gradient. Oh, okay, so we're going to stop there and we're going to go to the question part before we hit our xerophytes. Okay. So, if you open up your question pack, oh, there's a leaf on there.